All right, it's my pleasure to introduce Claudio. He is currently pursuing a PhD in veterinary medicine at the University of Sassari in Italy, focusing on granulomatous fish diseases and fish mycobacteriosis. Last year, he joined the Aquatic Animal Health Lab at UC Davis under Dr. Soto, working on the development of a live attenuated vaccine for fish mycobacteriosis and studying mycobacteria biofilm disinfectant susceptibility. Looking forward to your talk.
Hey, thank Nick for the kind presentation and thank AFS for letting me present. Today I will talk about susceptibility of mycobacterium species biofilm to common disinfectants used in aquaculture. And since I mentioned them, let's talk more about myco. And they are gram-positive, rose-shaped, acid-fast facultative intracellular pathogens that are categorized in tuberculosis and non-tuberculosis mycobacteria. This last group are also known as environmental bacteria, mycobacteria, and so far 193 species have been isolated. In this group, we also find strains that can be pathogenic to fish, with Mycobacterium marinum complex and Enchelonia abscessus complex as the most reported ones. These strains have trophies for both fresh and saltwater fish, and some of these strains also have zoonotic potential. In fish, the disease usually manifests as ulcerative skin lesions, exophthalmia, white nodules throughout intergal organs, leading to chronic infections with low but persistent mortality rate. As of today, no treatments or vaccine are available for this disease, and in aquaculture systems, prevention of outbreaks mainly relies on disinfection and eradication of infected stocks. So mycobacteria are kind of unique in the microbiology landscape, and this is due to their characteristics, starting from the cell wall where we have the mycomembrane formed by mycolic acid and glycolipids that is involved in structural integrity, permeability, resistance, acting as an impermeable barrier to antimicrobial agents, disinfectants, and antibiotics. The cell wall alongside the extracellular matrix contributes to the biofilm formation. Particularly, the extracellular matrix is required for retention of water, cell aggregation, and adhesion to surfaces. And for mycobacteria, the formation of biofilms sometimes leads to polymicrobial biofilms, where other microorganisms might be included, leading sometimes to co-infection. Lastly, biofilm provides a second layer of impermeability that can protect them from antimicrobial or disinfectants. And here, I just wanted to show the biofilm formation process from a review from Guzman and co-authors in 2021. And starting from the planktonic bacteria, the first step is this adsorption when they get in contact with the surface and starting having the first hydrophobic interactions. Then it is followed by the adhesion, where bacteria cells start to get in touch each other and start expressing some proteins as adhesins that strengthen the adhesion. And then they start to form microcolonies and where they start to grow and transitioning from a bidimensional to a three-dimensional form and producing the extracellular matrix. Then microcolonies can grow and become macrocolonies, leading to the maturation where planktonic cells might be included and aggregated to the biofilm, and here the biofilm start acting as a different kind of organism. Once the biofilm is mature, when a stressor event occurs, such as hypoxia or lower nutrient availability, it can undergo a remodulation that might culminate in a disgregation of the biofilm and some cells that can go and colonize other surfaces and places. What I wanted to highlight here is that the bacteria that are dispersed most of the time express more virulent and resistant phenotype since they undergone through some modifications that allow them to form and persist in the biofilm. And here I put some of the key functions of biofilm, starting from the protection, acting as a physical antibiotic resistant, nutrient retention and exchange, gene transfer between mycobacteria or other bacteria, colonization persistence, communication, and viromediation. However, some studies have been conducting addressing mycobacteria disinfectant susceptibility, but less work are present on biofilm mycobacteria disinfectant susceptibility. And as highlighted in this recent review, mycobacteria resistant to antibiotic and disinfectant has become a huge research topic that needs to be further investigated. And this is particularly true for the aquaculture systems where mycobacteria can lead to disease outbreaks and economic loss. And this kind of aimed our study that was to compare biofilm formation among different mycobacteria species previously recovered from outbreaks of mycobacteriosis in fish and investigate the resistance to common disinfectants used in aquaculture system. And to do so, we used isolates that were previously obtained from deceased fish and sourced from the culture collection at the Aquatic Animal Health Laboratory at UC Davis, and based on phylogenomics from Gupta and co-authors in 2018, we ran an RPOB characterization of our isolates. And as the phylogenetic tree shows, our isolates clustered along Mycobacteroides scheloni, Mycobacteroides salmonifilum, and Mycolicibacterium arquilense. For the rapid growers, Mycobacteria, and for the slow growers, we had Mycobacterium marinum. Then we used these isolates to characterize the biofilm formation using minimal bacteria eradication concentration assay, adapting methods from Heckman and Harrison. Basically, we prepared a 0.5 McFarland on BHI for all the isolates, and then we diluted them until obtaining a 1.5% to the 5 CFU per ml, and then aliquots of 150 microliter were distributed in a 1960 well MBEC plate that was covered with a PEG lid. This plate was incubated at 25 degrees Celsius at 110 RPM and incubated for 2, 4, 8, 12, and 14 days. At each respective time point, we rinsed for 10 seconds in PBS to eliminate non-adherent bacteria, and then moved to a bacteria growth check plate where the strains were sonicated to dislodge the biofilm, serially diluted, and then spot plated in triplicate in blood agar, incubated at 20 degrees Celsius for 7 to 10 days. And this is what we obtained. And as we can see, all the mycobacteria were able to form biofilm with higher bacteria biofilm associated for the fast-growing bacteria. And based on these results, we decided to use 12 days as the best biofilm formation time point. Something that I want to highlight here is that it was a little challenging to standardize the dose because some mycobacteria show clumping behavior, particularly for the M. marinum and M. arquilense, where the starting inoculum was 10 to the 4 CFU per ml. For the disinfectants, we use commonly disinfectants that are used in aquaculture system based on the review of Dr. Yano and co-authors in 2012. And in details, we used bleach 200 ppm for 30 minutes, Ovadine 50 ppm for 30 minutes, Virkon Aquatic 10 gram per liter for 15 minutes, 3% of hydrogen peroxide for 15 minutes, and lastly, 70% ethanol for both 30 and 15 minutes. And before the challenge, we matured 12 days mature biofilm that we evaluated with methods that I previously described. And this was the concentration of biofilm-associated bacteria that were then challenged with our disinfectants. And then for the disinfectants, we diluted them in a filter sterilized water from an aquaculture facility, and mature 12 days biofilm were then challenged for 30 minutes or 15 minutes, depending on the disinfectants, then were rinsed for 10 seconds in PBS, and the lid were transferred in a recovery plate to re-equilibrate for 30 minutes. Then 3 packs per strain were sonicated on high for 30 minutes, and the bacteria suspension, 100 microliter, were moved to the log reduction plate for serially dilution. Then they were spot plated in quadruplicate in blood agar, incubated at 28 degrees Celsius for 7 to 10 days. And this is what we obtained. And as we can see, for the 15 minutes treatment, which included Virkon, hydrogen peroxide, and ethanol 70%, it was effective only for Mycobacterium marinum, where for the 30 minutes treatment, which included bleach, Ovadine, ethanol 70%, it was effective for both M. marinum and M. arquilense, but didn't affect the M. cheloni and the M. salmonifilus strains. So moving to the discussion, all mycobacteria were able to form biofilms with differences among different species. Mycobacterium marinum was susceptible to all disinfectants, testings in accordance with similar results from another study. Mycobacterium arquilense was susceptible to bleach, Ovadine, and 70% ethanol only for the 30 minutes treatment. And unfortunately, none of the disinfectants were effective in eliminating M. cheloni and M. salmonifilus biofilm. This might be due to the formation and production of abundant extracellular polymer substance. So in conclusion, these preliminary results suggest that biofilm formation can vary among different mycobacteria species and highlight the need for effective disinfectant strategies in aquaculture facility. Resistance to disinfectants needs to be clarified, especially due to the widespread use of disinfectants, particularly, for example, after COVID-19 pandemic, and emerging scientific evidence on the importance of resistance of non-tuberculosis mycobacteria continues to be better clarified. For future steps, we can include more mycobacteria species pathogenic to fish and repeat the assay to increase the statistical significance, optimize the disinfection concentration and exposure times, even including other detergent or other disinfectants, and understanding the genetic and biochemical mechanisms underlying the biofilm formation and disinfectant resistance of these mycobacterium species. With this, I would like to acknowledge my supervisor, Dr. Soto, and all the people from Aquatic Animal Health Lab that I miss a lot, and my supporting institution, University of Sassari, and I'm open to questions if you have any.
Great presentation, Claudio.
Thank
Interesting
you.
project. We've got a minute for questions, folks. Feel free to drop them into the chat or unmute yourself. All right, well, you're getting compliments in the chat.
Oh, thank you.
Great presentation. Thank you very much.
Thank you.